As per the scientific explanation of homeopathy proposed by MIT or molecular imprints therapeutics, potentized medicines contain molecular imprints or intermolecular voids in hydrogen-bonded supramolecular clusters of water-ethanol molecules carrying the conformational imprints of drug molecules, which can act as artificial binding sites for pathogenic molecules, thereby removing the pathological molecular inhibitions underlying disease conditions. For validating this concept, we have a highly relevant peer-reviewed research paper before us by a Swiss chemist Louis Rey regarding thermoluminescence phenomenon of ultra-high dilutions of lithium chloride and sodium chloride, published in December 2002. This paper is available in its full format, Thermoluminescence of Ultra-High Dilutions of Lithium Chloride and Sodium Chloride. Physica A, 323, 2003, pp. 67-74. Although this work had been frequently quoted by many supporters of homeopathy earlier, nobody could understand its exact meaning and relevance due to their lack of a scientific perspective of homeopathy, which obviously led to wrong conclusions and interpretations. Actually, this historical work of Louis Rey was gravely discredited and humiliated before scientific community by pseudoscientific interpretations of energy medicine homeopaths. Hence it has become an immediate necessity to reinvent and reinterpret this research work using scientific MIT perspective. As per the experiments conducted by Louis Rey, the scientific advisor of Switzerland, it was shown that the presence of active ingredient alters the chemical bonding of water and the change is maintained even when the solution is diluted several times even above Avogadro limit. He used thermoluminescence, an indicator of the chemical structure of a substance, to prove the premise. As per the reported work, samples of homeopathic drugs lithium chloride 15C and sodium chloride 15C have been cooled down to 77 K, and irradiated by X and gamma rays, then progressively re-warmed to room temperature. During that phase, their thermoluminance has been studied and it was found that, despite the dilution beyond the Avogadro number, the emitted light was specific of the original salts dissolved initially. This wonderful observation that high dilutions of salts very much above Avogadro number retains the specificity of thermoluminescence patterns reminding of original salts seems to be very crucial. This phenomenon obviously indicates some sorts of supramolecular rearrangements of medium as well as formation of nanostructures. These nanostructures formed of water-ethanol molecules could be explained only in terms of molecular imprints or intermolecular voids carrying the three-dimensional conformational details of individual molecules of salts, as hypothesized by MIT concepts. Thermoluminescence studies have been developed and utilized so far as a tool to study the structure of solids, mainly ordered crystals. In the present study, the researchers successfully utilized it in ultra-high aqueous dilutions, which demonstrates the short-range crystalline character of water as well as high-dilution preparations. Actually, the researchers took up this work to challenge the water memory theory, but proved it otherwise. They confess in their report that they designed and executed the project since they thought that it would be of interest to challenge the water memory theory according which pre-existent structures in the original liquid, developed around some added chemicals, could survive a great number of successive dilutions when done under vigorous mechanical stirring. Another important point to be noted is that the researchers did not use commercial samples as most researchers do, but prepared themselves 15C dilutions of lithium chloride and sodium chloride under the guidance of Boiron Labs. This fact provides more scientific credence to this study. The study showed quite clearly that the initial addition of the solutes in the original potentizing medium leaves a permanent effect even when, by successive dilutions made under strong vibration, all traces of solute have disappeared. Authors say, the results were reproduced in several repeated experiments, beyond any ambiguity. Thermally stimulated luminescence, often called thermoluminescence, is a well-known phenomenon amongst the thermally stimulated processes such as thermally stimulated conductivity, thermally stimulated electron emission, thermogravimetry, differential thermal analysis, differential scanning calorimetry, etc. Its theory and applications have been fully developed inter alia by McKeever, Chen and Visacekis, and it proved to be a most interesting tool to study the structure of solids, mainly ordered crystals. To that end, the studied material is activated at low temperature, usually by radiant energy such as UV rays, X-rays, gamma rays, electron beams, or neutrons, which most generally creates electron-holes pairs which become separately trapped at different energy levels. Then, when the irradiated material is warmed up, the heating serves as a trigger to release the initially accumulated energy, and the trapped electrons and holes move and recombine. A characteristic glow is emitted most often under the shape of different successive peaks according to the depths of the initial traps. As a general rule, this phenomenon is observed in ordered crystals, though it can be equally seen in disordered materials such as glasses. In that mechanism, imperfections in the lattice play a major role and are considered to be the place where luminance centers appear. Thus, luminescence is a good tool to study these imperfections and understand how they appear in the crystal. This is exactly along those lines that the researchers carried our first investigations, starting, this time, from liquids which were turned into stable solids by low-temperature cooling. Working essentially with water, they have shown that the luminescence glow of irradiated hexagonal ice consisted in two major peak areas, peak 1 near 120 K and peak 2 near 166 K, having well-defined emission spectra, the D2O samples giving a much higher signal than the H2O ones. In both cases, unirradiated samples gave no signals whatsoever. For both D2O and H2O, it was shown that the relative intensity of the luminescence glow was a function of the irradiation dose, and that at least for peak 2, it did show a maximum between 1 and 10 kGy. As a first hypothesis on the nature of the emission itself, it has been suggested by Teixeira that peak 2 could be connected to the hydrogen bond network within the ice, which, in turn, could result from the structure of the original liquid sample, whilst peak 1 looked to be closely related to the molecule. This strengthens the views on the involvement of hydrogen bonds in this mechanism. To develop this concept further, the researchers did select to study the effect of lithium chloride on the luminescence of irradiated D2O ice, since this particular substance is known to suppress hydrogen bonds. The result, indeed, is spectacular and, at the relatively low concentration of 0 to 1 m, peak 2 is totally erased, whereas the basic emission of peak 1 remains almost unchanged. At that point, the researchers thought that it would be of interest to challenge the theory according which pre-existent structures, in the original liquid, developed around some added chemicals, could survive a great number of successive dilutions when done under vigorous mechanical stirring. To that end they prepared, courtesy of the homeopathy manufacturers Boiron Laboratories, ultra-high dilutions of lithium chloride and sodium chloride by successive dilutions to the hundredths, all done under vigorous mechanical stirring until they reached at the 15th dilution. A reference sample of solvent alone was also prepared according to this technique. They did proceed, then, to the activation of these materials by irradiation according the following experimental protocol. 1 cubic centimeter of each solution is placed in aluminium test cavities of 20 mm diameter and 2 mm depth and frozen to 20 C on a cold metallic block. The frozen systems are kept 24H at 20 C to achieve stability into their crystallization pattern, and they are immersed into liquid nitrogen and kept at 196 C for 24H. In a first set of experiments, the frozen ice discs are irradiated at 77 K with 100 kV X-rays to achieve a dose of 0 to 4 kGy. 30-minute previous determinations were done to check that the discs having identical positions in the field did receive the same dose. Dosimetry has been done using Harwell, FWT, and alanine dosimeters. After irradiation, all the activated samples are transferred into a liquid nitrogen container and kept there for a week time, to even out whatever small differences could exist between them. Finally, All samples are placed in the luminescence equipment and their respective glow recorded with both a photomultiplier and a CCD camera connected to a spectrograph in the course of rewarming 3min between 77 and 13K, as has been done in our previous published experiments. Much to their surprise, the experimental results do show without any ambiguity that for an X-ray dose of 0 to 4 kGy the luminescence glows of the three systems were substantially different. These findings did prove to be reproducible in the course of many different identical experiments. To compare the curves between them, the researchers normalized the emitted light readings, taking peak 1 as the reference. In doing so, we obtain for peak 2 the different curves presented, which show quite clearly that the initial addition of a solute sodium chloride and lithium chloride in the original solvent leaves a permanent effect even when, by successive dilutions made under strong vibration, all traces of solute have disappeared. More remarkable were the fact that, by far, lithium chloride demonstrates a stronger hydrogen bond suppressing ghost effect, which could be related to the larger size of the lithium ion. A second set of experiments done with gamma rays courtesy of CELESTIN reactor, COGEMA, Markle, at a higher dose 19 kGy did confirm these findings. It appears, therefore, that the structural state of a solution made in water can be modified by the addition of selected solutes like lithium chloride and sodium chloride. This modification remains even when the initial molecules have disappeared, and the effect is the same at different irradiation doses, and for different radiant sources such as X-rays and gamma rays. As a working hypothesis, the researchers propose that this phenomenon results from a marked structural change in the hydrogen bond network initiated at the onset by the presence of the dissolved ions and maintained in the course of the dilution process, probably thanks to the successive vigorous mechanical stirrings. Since the researchers had no idea of molecular imprinting, they proposes that this phenomenon results from a marked structural change in the hydrogen bond network initiated at the onset by the presence of the dissolved ions and maintained in the course of the dilution process, probably thanks to the successive vigorous mechanical stirrings. C. This hypothesis comes very close to the concept of molecular imprinting. If we fail to explain the observations of this monumental research in terms of molecular imprinting, there remains the danger that it will be hijacked by energy medicine theoreticians, by interpreting in terms of dynamic energy, vibrations and the like. Actually, energy medicine homeopath Jan Scholten has already done such an exercise, by saying information of drugs imprinted in water are the cause of luminescence observed by the researchers. Then he very cleverly fits this luminescence into his energy medicine framework of bioluminance, vibrations, vital force, resonance and other pseudoscientific theories being part of unscientific theoretical system of homeopathy. To be specific, precise and fitting to modern scientific knowledge system and its accepted paradigms, this phenomenon of luminescence of ultra-high dilutions demonstrated by researchers should be explained in terms of molecular imprints of original drug molecules formed in ultra-high dilutions. Such an interpretation of this important research will strongly ratify our MIT concept of molecular imprints in homeopathy drugs that produce therapeutic effects by acting as artificial binding sites for pathogenic molecules.